Human brains are wildly complex, all these interconnected parts, and each one is relying on signals from other parts to know what it's supposed to do, how it's supposed to grow, and whether it's running properly. Up till now, we've been able to study animal brains. We can look at post-mortem human brains. The living prefer mostly to not have their brains sliced open. But now we have one neuroscientist at the College of Medicine who's shown promising results for neurodegenerative medicine, growing organoids, miniature human brains. Dr. Tyler Wenzel is our guest today on Researchers Under the Scope. Hi, I'm your host, Jen Cannell. Today is Thursday. It's the 21st of May. And today we are looking at brains, specifically the brains of children and teenagers and the brains that are still under construction and kind of in development as rare degenerative diseases attack. We're also looking at why Dr. Tyler Wenzel's human brain organoids, these sort of synthetic mini brains, why those may now give us a far clearer picture of the treatments that are and are not effective. Dr. Tyler Wenzel is an assistant professor of psychiatry at the University of Saskatchewan's College of Medicine, and they join us now. Welcome to the podcast, Tyler.
Thanks for having me. I look forward to this talk.
Okay, brains, and specifically young, sort of developing brains. Where did your first interest in this branch of neuroscience come from?
I used to be a high school teacher, and I would teach biology 12. And in biology 12, you learn a lot about the different body systems. You learn that the heart goes a lub-dub and blood goes around the body. My brain can wrap around that.
Sure.
You know, when you're breathing, your lungs, you inhale, they get bigger, air goes in, air goes somewhere, but I'm breathing and I'm alive.
Yeah.
Textbook explained it beautifully. I could understand it. But then they got to the brain section. And the brain section, for those who don't remember Bio 12, it essentially says, so there's some electrical stuff happening there, and that makes you think and move and do things.
But we don't really know how.
We have no idea how. And I'm like, okay, I'm a robot who has, blood and stuff, but I didn't understand it. And when you're talking with students, they're also like, how does that work? And I'm like, that's a good question. I have no idea. So from there, I was just fascinated by the brain because it's the one part that I couldn't understand. And, very long story short, at one point during my life, I had the opportunity to go and get a master's and a PhD degree in neuroscience. And the rest is really history, but it all really started just from being like, this is the most underdeveloped part of the biology textbook.
It's really hard to study a human brain. Yeah. People don't like it if you cut into them.
No.
They really don't. It's not helpful most of the time. And people will refer to it as the supercomputer or the black box. But when it comes to actually testing medicine. I remember when we were talking anesthesiology, Dr. Peter Hedlund saying, yeah, nobody really knows why these medicines do what they do. And that's frustrating me. And I've got to get to the bottom of some of this. So during your master's, during your PhD, one of the things you proved is people are not rodents. Yes. People are not rats. People are not mice. Why did you look so hard at that? And why?
When you're doing your PhD, you have to discover something. And as I was doing all these experiments, I was essentially discovered two separate things that said the exact opposite. And I was trying to understand why. And everyone was pointing at me being like, okay, you have these sets of experiments that say everything go down. And you have these set of experiments that say everything go up.
Uh-oh.
And
can they be replicated? Sure, I guess.
Yeah, I could replicate it. No one had any issue with the methods. They're just like, but why? And really what we came down to realizing is all of the thing that go down was from a mouse, and all the thing that go up was from a human.
Oh, that's a pretty significant difference.
It's really significant.
Like we're two different animals.
Yeah, almost like we have 37 million years of evolution between being a mouse and a human that could completely change how our brains work.
But when we test medicine for brains, we've tested it on rodents.
Yes.
What are the most profound differences that stood out to you after looking at this about why theirs go down, humans go up?
The most profound difference? There's many profound differences, but in the brain specifically, the brain, well, a lot of people don't know it has an immune system. People just think the immune system's in your blood. And the way that the mouse brain immune system works is completely different. It's super responsive to any sort of infection, any sort of toxin, whereas the human brain is a little more resilient. It can kind of fight it off and not care as much. And that's really important for a lot of different brain diseases.
Right. Okay. So, and that's the glia? Like, what is it in my brain that creates, it's like the equivalent of white immune cells, right? Like white blood cells.
Not quite the white immune cells, but the macrophages. So the immune cells in your blood that go around eating things. You have those in your brain too, and they're called microglia.
Microglia. Okay. So continuing on with that, you come through, you do your postdoc work, you ended up here in Saskatchewan. How'd that happen?
So how that happened was what I always say is just serendipity and opportunity coming together. So at the end of my PhD, I had these two different discoveries, which basically amalgamated in the fact that humans and mice are different. So if I'm going to study human disease, I want to work with human tissues, human cells, and figure that out. Now, around this time that I finished my PhD, there was becoming a more widely adopted tool that researchers could use, and they're pluripotent stem cells.
Yes. Okay. For anybody who last heard about stem cells like back in the 1990s and was like, ew, we're using fetal tissue, that's not cool. What are pluripotent stem cells?
So pluripotent stem cells, by definition, I'll start with that first. They are cells that if you give them the correct signals, they can turn into any cell type. So from pluripotent stem cells, this is ultimately how I make a brain organoid or a mini brain, because I give it the correct signals. I can also make the specific cell types of the brain too.
What's its source material?
So its source material for that is not fetal tissue. We don't need that anymore. These cells can basically come from anything now. So genetic engineering has gotten really, really good. I'll leave it at that. But we can get pluripotent stem cells from skin. There are some people who you can get it from urine because there's cells in your urine, which is surprising to me. I've also made pluripotent stem cells from blood. So you can get it from effectively any sort of source tissue. And
your pluripotent stem cells would be different from mine. Mine have all my coding on them. Yours have all yours.
Yes.
So you can personalize this person's stem cell. Okay.
Yeah, correct. So if I grab, right now I'm healthy. So if I grab my cells, I will get pluripotent stem cells that have my genetic information. And if I make a brain from that, a mini brain, it'll be a genetic copy of my brain and will exhibit the things of my brain. If you do that from someone with some sort of disease, if it's a genetic disease, the pluripotent stem cell will have all that genetic information. So if you make any sort of brain tissue from that, that brain tissue will also have the pathology of that disease.
How long does it take to make to grow all these different parts of a synthetic human brain?
So it takes about, you get kind of a brain structure where you start having electrons, electrical activity. That's about 30 days. It's not very long, but keep in mind, everyone that these are small brains. They're mini. I call them mini for a reason. They're about 2 millimeters in size. They're not a full entire brain.
I can't hold them in my hand. I can't even see it hardly. So they exist like in what kind of media?
Basically what we did is we made a media that just, it's a vat of nutrients that your cells need. Like there's nothing special or unique about it, really. It's just a media that mimics what your brain is floating in.
From there, if you have, okay, you probably do at least a couple of them. What does that change about testing different treatments and different pharmaceuticals or different combinations of things on my brain? What does it mean you can do now?
So it means there's many different things you can do with this. So
many.
So one of them, and this is not the work that I generally focus on right now, but if you had, say, a treatment resistant disorder, there's treatment resistant schizophrenia, treatment resistant depression, stuff like that. In theory, if that is genetic based, which in schizophrenia it is, you could grow a mini brain and see which drug works on your treatment resistant disease.
Oh.
So that is one unique aspect. Where I kind of fall in more under is I work on genetic diseases of youth and children. And so we basically will either take cells from a patient with a brain disease, we will turn them into pluripotent stem cells, and we will make their mini brain. And because it has their genetic information, we get a brain that has their pathology. Now, to link this back to when I was saying mice and humans are different.
Yeah.
If you do that same mutation often in a mouse, the pathology won't appear.
What? Oh.
So it allows us to actually get a pathology, a human-specific pathology that we can actually study.
And replicate. It doesn't render mice completely useless for us, but let's just put them to the side. Yeah. Different species. And what's beautiful about this is because we can't just see inside my brain and all the electrical wiring that's happening in there and the parts that are lighting up. We can measure some of it. But this would allow you to do so much because it's not just a collection of neurons you're looking at, isn't it? There are other kinds of...
There are other cells, there's a lot of cells that keep the neurons alive and functioning correctly.
Their whole support system is big. How did you do that? Like, how have we figured that out?
A lot of trial and error. Like, if I showed you my laboratory notebook, it is just systematically going through a bunch of different nutrients, adding them at different time points, and eventually being like, well, that one worked.
Really?
That was 3 years of my life.
Oh, my goodness. Were those the pandemic years?
Yes. I had nothing else to do.
So I thought I'd replicate the human brain. Yeah. Worked out. What the heck? This is crazy cool. Yeah. And how do you not build in mistakes? Like, how do you make sure that, okay, this is as realistic as we can get without having coding problems?
So this is something where I will admit it is as close as we can get to the human brain. There are still some differences between this and a human brain. So one of the challenges when your brain's growing, and you're from a little fetus and you're growing and they eventually become a human, is that your body works a lot on gradients. So, there's a lot of molecules on the right end and there's a lot less molecules on the left end. And there's kind of this gradual increase. Right. And those are very tightly regulated. And that's how you get the brain that you and I have. If you look at a brain picture, you have like a cerebellum in the back. It kind of looks, stripy. You have your big human cerebral cortex. Your brain has a bunch of different structures in it. Yeah. And those are all developed over essentially gradients. So when we grow these mini brains in a petri dish, we have a lot less control over those gradients. So we get as close as we can. But while, your brain and my brain, if we happen to take them out, I don't recommend that, but if we do,
I won't,
they will look very similar. Our cerebellums will be in the same place. Our cortexes will be in the same place.
Okay.
in these mini brains, you don't really have that tight structure where you know where everything is. It's kind of just in there somewhere.
Sort of, I'm picturing a blob more than disparate structures.
It's very much a blob, and if you want to like imagine in your head what it looks like to your naked eye, it looks like if you took a piece of chewing gum, like a white piece of chewing gum, you chewed it up, and then you kind of like spat it out and kind of smoothed it out a bit. That's literally what it looks like.
That's the mini brain.
Yeah.
And I know we hired you here, brought you in at the University of Saskatchewan. This would be going back to October 1st, 2024.
Yeah.
And at that point, you didn't have a full setup lab. You knew you had made some progress with the mini brains, and they were very excited to have you. So from there to here, how did you manage to build your lab?
Probably the most unorthodox way possible. I'm someone who understands like research budgets. They're very small. I also really appreciate that a lot of our money comes from either donors, the federal government, which is ultimately taxpayer dollars, or it comes from, the Saskatchewan Health Research Foundation, also provincial dollars, which are tax dollars. So to be conscious of that, I always try to be as budget conscious as possible. So when I got hired, there was a very unique situation that happened. To set this up, when you work in stem cells, a lot of the stem cell industry across Canada is businesses. So a lot of people in my field don't actually work at universities. They work at corporations or at startup companies and stuff like that.
At private industry.
At private industry. And so those are a lot of my friends.
Ah, cool. I'm guessing you met them at conferences.
Or I worked with them while I was doing my PhD. Or my friend of a friend of a friend worked with them while they were doing their degree.
Oh, okay.
So it's a very small group, but it's an in crowd in a way. And the key word I said there is startups. And some startups don't work. Or sometimes you have a big corporation who's like, we want to put a lot of investment in this building, in this province. And then eventually, when business goes away, they might downsize.
Okay.
So what this means in my specific field is that a lot of companies often just say, hey, we have a lot of equipment to sell. And so I had a friend around the time I was getting hired who was like, hey, so we're downsizing our entire laboratory section of our business. And would you like anything? And I said, I didn't even ask what they had. I just said yes. And I was like, please hold on to everything. I can almost guarantee you I'm going to buy everything that you have. I was out of town at the time. Anyway, when I came back into town, I went to their office and I said, okay, what do you have? And they said, here it is on this very long table. And it was, if you price it out all brand new, it's very easily over 6 figures.
We're talking centrifuges.
We had three centrifuges. We had multiple analytical balances, which are super sensitive scales to weigh stuff. We had pH meters. We had freezers. We had fridges. We had all the basic equipment that basically is like, you just take that and you can start 3 laboratories with it.
And you said, sure.
I said, sure. And I said, how much? And they said, well, does $5,000 sound reasonable? And I said, here's my checkbook.
5,000?
5,000 for over.
Well, it could be more than $1 million.
Yeah, if I went on eBay, I don't know if I need this job anymore.
Oh my goodness.
I do want this job, but
oh, good, because we need you still. We just got you. But that takes you through, and because usually you'd have to spend so much time applying for grant money, and I have to prove that I need this piece of equipment, and you sort of speed ran that.
Yeah. I am so grateful for my friends and my colleagues who let me do that, because I can, like, all the money that we started off with, it is literally going to just the research team and directly to the research. A lot of the equipment was all paid for.
Oh, that is so cool. And because part of it is you make, if you were a baker, I'd say you bake things from scratch.
Yes.
You don't use premix.
No.
Why does that matter?
Some people would tell me it doesn't, and I would tell them that it really, really does, because I learned science as a high school teacher. And in high school, you learn about the scientific method. And so the scientific method is a very strict set of rules that you follow, and everything has to be reproducible, and you have to know every single component. Like if there's any high school kids watching, if you had to do their bio 12 or chemistry 12 lab report, they're literally writing, okay, I added this amount and all this.
Yep.
So that's how I was trained.
Right.
And one thing I could never really adapt to going into university is a lot of university research groups use premixes. They will use pre-combinations of various molecules to do things. But they don't tell you what's in it, and they don't tell you how much. They just say, trust us, we made something that resembles kind of what you need.
But how are you supposed to reproduce that?
Well, you trust that they can remake it again, which is not how I was taught the scientific method.
I don't know if you passed grade 12 science with that. I can't copy off your neighbor. Yeah. No, but it also can very much affect your results in very sensitive situations, because you said those brain cells that we have resilient, they sort of take things in a little bit more than those rodent brains, but like you need to have them in a pretty neutral state to be able to test anything.
But also, if I add to that's what's very important is to make a brain, it also involves really making sure that everything is the correct concentration at the correct time, at the correct location. And, so if you have someone over in some sort of company who's making your thing and they accidentally add like 3 more little specks of powder, that could completely change your entire batch of brains that you make with that.
Yeah. And all your results and everything coming from it. When it comes to the diseases that you're looking at, typically not ones we see that often, youth degenerative brain disease. Yeah. How does it manifest in like children or teenagers?
I will say that there's a bunch of different degenerative brain diseases. The one that I'm focusing on right now is, it's called X-linked adrenoleukodystrophy. You've never heard of that ever. And because that sounds like a mouthful, trust me, it is if you try to say it. So I'm just going to
X-linked adrenoleukodystrophy. This is hard.
So for everyone listening, I'm just going to abbreviate it to ALD.
Perfect.
And a lot of the degenerative diseases for youth are an enigma, especially this one. So I kind of have to set it up, if you don't mind.
Go for it.
And so this disease specifically has multiple different ways that it can manifest. There is one that is kind of results in a steroid imbalance. So you can go see an endocrinologist who will give you some steroids and, everything will kind of work out and you'll be fine. Okay. Not super chill. It's not super chill, but, comparatively to where I'm going, it's chill, relatively chill.
Okay.
And so then the next one you have is if you have this disease, this ALD form, the disease might then say when you're between the age of, let's say, 40 and 60, you might start losing control of your limbs.
Okay. Like sort of spastic movements
stuff like that.
Okay.
And that's another form. Okay, you can still live. It's kind of uncomfortable, but then you have a third form. And this is the very serious form, and it's the cerebral form of ALD. And this is one which is the rapid degeneration of your brain. And I don't know if you know this, we need our brains to live.
Yeah.
So the rapid degeneration of your brain is no bueno.
No, how rapid?
So generally speaking, if you see symptoms, your prognosis is about 2 to 4 years.
Oh, no.
Yeah.
Oh, no. Does it take out your spinal cord too, or more just brain?
It can affect your spinal cord, but really the thing that we're really focusing on is the brain, because that's what makes you breathe and that's what makes you think. And
what are the symptoms? Like, how do you know that a kid's got this? Because I can imagine it mimics so many others.
Yeah. So kind of how this works is we know this disease results in a certain mutation in your gene, but that mutation can cause any one of the 3 different forms that I just mentioned. And it's a little more complicated than that, and this is why I love the disease personally, is that there were twin studies done where, and for those who don't know, identical twins have the exact same genome.
Yeah. So every chromosome is the same.
Every chromosome is the same. Every gene's the same. So if they have a mutation, it's the same mutation.
Right.
And there is studies done where one twin with the mutation will get the cerebral form of ALD, and the other twin will be asymptomatic for their entire life.
What?
Which kind of flips on its head like everything we know about how genetics work and how genetic disorders work.
Thought I was good. But like, why doesn't the other twin get it? We just don't know, or it's chance.
We have no idea, because you would think that them being identical twins, they would grow up in the same environment, exposed to the same things. So, if one randomly, I don't know, was living in a house with black mold, I don't know why that came to my mind. The other one would live in a house with black mold.
Right. Or one's living near radiation. The other is, the other would be too. So even in identical twins, it doesn't present the same way.
Doesn't present the same way.
Does it take away motor skills, or what does it do to a person?
It can do, the disease?
Yeah.
The disease can do anything from nothing for your entire life to
attack your brain quickly and you're dead in 2 years?
And you're dead in 2 years.
This is horrible. How do we treat it right now?
So right now, you undergo a procedure. It's called a hematopoietic stem cell transplant. Big wordy thing. Some people might think of a bone marrow transplant.
Yeah, this sounds like cancer treatments where you have to get chemo first or radiation.
That's exactly what it is. So the treatment is essentially the equivalent of treating leukemia. So you get chemotherapy or some sort of irradiation. It'll make a bunch of holes throughout your body. What those holes do is they leave the opportunity for other things to be put in those holes. So a lot of the time you're putting other people's immune cells into your body to fill those holes. And for some reason, when you have other people's immune cells into your body, that will cure a lot of different diseases.
If they match well enough, and if you take immunosuppressants your entire life.
Yeah, you have to take immunosuppressants your entire life. The mortality for that procedure is between 5 to 20%, and that number just increases with age.
And that's the best treatment we have?
That's the best treatment you have.
Okay, what do you have?
So I have a way to do a very similar treatment without needing to do radiation or chemotherapy. It only works for brain diseases. I won't get into why. If anyone wants to, hit me up on my email. But it only works for brain diseases. And we've basically designed a method where we can create the holes necessary in your brain or in your spinal cord selectively, because chemotherapy and irradiation makes holes throughout your whole body.
Yeah.
So we can do it in just your brain, just your spinal cord, and then we can inject the immune cells specifically into those locations. The way we do that eliminates many of the things that make chemotherapy uncomfortable, many of the things that make any sort of stem cell transplant uncomfortable. We don't need to do any of that.
So less long-term, invasive, harsh.
Yes.
Could you ever do it with my own cells if things go well with where you're going with organoids? Could you ever take my own cells and basically bring them back to health?
Yes.
And then put them in the holes?
Yes. So there's two different things we can do. And immunology is in this crazy place we can do right now.
That's cool.
So if you had ALD, we can take your cells out. We can genetically correct the ALD mutation you have. And then if we put those corrected cells back into your brain and spinal cord, you could basically live more or less a completely happy life. You might want to see a doctor every once in a while just to make sure, because things can go weird, but you're more or less living a happy life and you don't have to go through chemo.
Wow.
There are other really cool things that we could also do. And this is a lot of folks over in Japan have kind of developed this. Believe it or not, you know how you might hear that when you need to get like a blood donation, you need to have matching.
The closer you can match, the better.
The closer you can match, the better.
Yeah.
So there's a group in Japan who spent a lot of time and a lot of research and a lot of money, and they basically found that there are 5 pluripotent stem cells lines that can match to 99% of the human population.
Fantastic.
So with that, we might even not need to use your own cells. We could just grab these cells off the shelf.
We'll call them the generics. They go for everyone.
Yeah. And just give those to you. So that would actually reduce the cost of the treatment because they're generics and they shouldn't have any rejection potential.
This sounds like space medicine at this point, but how close is this to reality? Because sometimes you do get to see clinicians who have patients with this condition, with a number of neurodegenerative conditions. Like how much?
So what I'm talking about is, it sounds like science fiction. I often start all my lectures saying, so what I do is science fiction, but it's also reality. And so there are treatments like this that are currently being used right now. But when I say that, I always have to add an extra bit of caution. So these treatments, you can, for example, you can get them in Canada, you can get them in the US. But I always caution everyone listening, if you go onto Google and you type in stem cell transplantation, you will get a lot of links that are not associated with hospitals. They're private clinics. And those stem cell transplants are not Health Canada or FDA approved procedures. And we might not know what they're injecting into you. So there's a lot of, in pop culture, there's a lot of athletes who you say, go to Mexico or go to, I think there's one in Vancouver now, and they'll get a stem cell transplant. Those are a completely different beast. They don't work. They're unregulated. They will take your $50,000 or more
sure.
gladly. But that's a different thing.
Results may vary.
Yes.
But yeah, the Health Canada approved ones, the ones that are available for patients here in Canada, even the odd patient that you would come across in Saskatoon, this sort of opens up what we can do to help them.
Yes. So the thing that we're trying to add to that paradigm is the direct brain targeted thing to circumvent the need for chemotherapy.
That would be fantastic. And this seems like if it works in this particular genetic disease for young people, it would work in other gene related brain issues.
Yeah. While we focus on ALD, that's kind of like our starting point, our proof of concept. Once we have shown it in ALD, for example, youth, pediatric brain disorder that can be treated by a stem cell transplant, our procedure should be able to be used in that too.
Wow. That's fantastic. And Tyler, thank you so much for even just walking me through this, because it is like watching a window into the future. This is so cool. Thank you for spending all those pandemic years figuring it out.
Thank you for the pandemic happening to give me the time to do it.
Upsides. Really, there are upsides.
You always got to look at the silver lining.
Exactly. Yeah, no, thank you for being on the podcast. I'm sure to appreciate it.
I would like to thank everyone who literally made the research possible. Without the support of the Saskatchewan Health Research Foundation, the Natural Science and Engineering Council of Canada, NSERC, and actually couldn't even start this project. Like, I actually needed those two things to come together to even start. And there was no other way to start. And even recently, I would like to thank the Stem Cell Network, because they basically put a giant spotlight on what we're doing in Saskatchewan. So I thank them for the award, the Rising Star Award they just gave me.
I'm really, really glad that the serendipity and the luck and whatever it is that you brought here in the pandemic know-how, that it all did come together this way. And I just hope we can keep you as long as possible. That would be super cool. Thank you.
Thank you.
Dr. Tyler Wenzel is an assistant professor of psychiatry at the University of Saskatchewan's College of Medicine. His research focuses on youth brain health and coming up with better ways to treat children and teens who have degenerative brain disease. You can learn more about his brain organoids and more about the Wenzel lab at neuroscience.usask.ca. And I'll drop those links and Tyler's email into our show notes. Researchers Under the Scope is a presentation of the Office of the Vice Deans of Research at the University of Saskatchewan College of Medicine. We record and produce this podcast on Treaty 6 territory. We pay our respects to the First Nations and Métis ancestors of this place. We reaffirm our relationship with one another. And we'd sure like to thank you for listening and for paying attention to science. I'm Jen Cannell. Thanks for being with us here on Researchers Under the Scope.